Hello, my name is Coleman Berry, and for Genetics 311 Exam Mini Video 4-1, I'm going to be talking to you about the 4 different types of point mutations that can happen in a DNA sequence. One may ask, what exactly is a point mutation? A point mutation is a mutation that only affects one or a very limited number of nucleotides in a genetic sequence. This causes a substitution of one base for another. Point mutations can cause a multitude of effects on the protein that is produced by the DNA. Point mutations happen via 2 methods, transition or transversion. Remember, pyrimidine bases are A and G, and purine bases are C and T. Transition is when a pyrimine base is substituted for another pyrimine base, or when a purine base is substituted for another purine base. Transversion is when a purine base is substituted for a pyrimide base, or when a pyrimidine base is substituted for a purine base. First, we have the original DNA strand. It is then translated into mRNA and then amino acids that will eventually create proteins. The first mutation is a missense mutation. In this case, a guanine was substituted for a thymine, therefore making it a transversion mutation. The strand is then transcribed and becomes a uracil. The original amino acid was trophonin, and then the translation caused a change into an amino acid to cytosine. This change in the amino acid is known as the missense mutation. If this does not affect protein function, it is considered neutral. Next, I will discuss a nonsense mutation. In this case, a guanine was substituted for adenine. This was a transition mutation because the adenine did not transcribe into anything else, which leads to the stop codon. The third type is a silent mutation. Here, a cytosine was substituted for a thymine. This substitution makes it a transition mutation. The scribe is then transcribed and the thymine becomes an adenine. This causes a translation of the amino acid asparagine, but it does not change since it was already asparagine. There is no amino acid or protein change. Finally, a read-through mutation. An adenine was substituted for a cytosine, therefore this is a transversion mutation. Through transcription, this cytosine does not change. The stop codon was taken away and then translated into the amino acid cytosine. The longer protein chain makes this a read-through mutation since the stop codon was removed.